With that, I just want to introduce Benno Schwikowski, who is one of the co-founders of Cytoscape. He's based at Institut Pasteur in Paris, and he's going to be talking about visual analytics and systems biology. So thanks, Benno.
Thanks a lot, Manny, for inviting me to this meeting. It's exciting to see what's going on in this project that has many links with the things that we're doing, and I'm very happy to tell a little bit about my viewpoint about these things, how these things are connected. I don't have balloons or anything. I just changed my title on you, but just a little. So it's still about visualization and what we use it for. I would like in my talk to highlight some thoughts that are maybe non-obvious, I hope, to some of you, so that you can take additional message home about the relationship of visualization and complexity in terms of the biology that are non-obvious, and tell a story about computational complexity and what it has to do for us with visualization. So just very briefly, the general sort of framework in which we view our work is this one, in which we are trying to use many times network models, but also other types of models, in order to connect the molecular measurements that we can make, often on the low levels or medium levels in the hierarchy of biological complexity, to connect them to phenotypes that matter. And these questions that in many cases clinicians, but other scientists, maybe more fundamental researchers, motivate us to think about how can we make these connections, how can we interpret the data that we get on these different levels in a coherent way. And networks are one major way for us. And because we are invested since the beginning of the Cytoscape project into visualization and allowing human access to this data, not only bioinformatics and models that are pre-cooked, but also try to be inspired by what we can actually see in a data-driven way. And these things really go together for us. So in my talk, I want to talk about the link of visualization to biological complexity, maybe somewhat idiosyncratic view on where the biological complexity comes from. I think there's something to be said there, that in current systems biology there's a way to characterize it as useful for visualization. And then talk a little bit about computational complexity and how it also, what it has to do with visualization for us in different ways, and how we try to deal with this. So the first part is going to be a little bit more fundamental and maybe adds a new perspective, and the second part is going to contain some of our current work that has to do with computational complexity, and provides an example of how we connect visualization and computational complexity. So complexity in general is well, it's maybe not the absence of simplicity, but we know, I think in a very generic way, we know that there are things that we cannot put together, that we don't understand, that we cannot interpret in a common conceptual framework together, and we're trying to develop formal models and mathematical and computational analysis to help to summarize, understand, and predict. In many cases, the models are not predictive, but visualization serves as an entry point, maybe to direct the attention of researchers to particular molecules, or we all know how these things go in practice right now, at least when it comes to large-scale data. Then there's, of course, smaller scale data in which we have stronger ideas about how the systems work and so on, and we can start writing equations and so on. Our group is mainly working on problems in which we don't even have these ideas yet. We don't have a pathway that is known to be the major responsible pathway for a particular phenomenon. And I'm not, excuse me if you're interested in the clinics or in the clinical aspects or the biology, I'm not going to talk a lot about that, but more of the connection between visualization and complexity, since I know that also most of you are with a computational background. But still, I think the perspective is important on what is it that we're actually trying to do. And there, for the occasion, I wrote down a postulate, namely that systems biology complexity can be decomposed into a number of specific different components, and there's something that follows from that for visualization. I like to show you 4 different ways that, to my mind, are really, in some sense, independent and that come together in real applications of this, that generate this complexity that in the end we would like to tackle using computational tools. So one thing for sure, and that's not a surprise to you, is just that we have these large networks in which molecules interact, and this is not only hard to draw properly, but it's also hard then to make any sense out of that. We all know that. I said yesterday the first name that we had for a cytoscape was hairball. Luckily, we had other names in mind, including that. But even when you look a little bit closer into these networks, it's very difficult, both from an experimental point of view and from a conceptual point of view. Here I'm showing you actually an old paper from Hamid Boulouri and co-workers Eric Davidson, in which they teased apart the regulation of the endo-16 gene in sea urchins. And what you see here is a linear representation of the DNA upstream of the endo-16 coding region. Upstream, you have these binding sites for different promoters. And down here, what they've done is, in painstaking work for many years, they've teased apart how the different transcription factors that bind at these different sites will in the end combine their quantitative impact, their activity that they have when they bind, to a value that allows you some prediction or modeling of the transcript level of this gene. Yeah. This is in eukaryotes. It's a lot simpler in prokaryotes, but still, I think it's an interesting example what happens when you go a little closer, even when you look at these large networks and can nicely draw them, or even in a small network that has only a few players, things are not necessarily simple. Yeah, and you can express this equivalently, or maybe even in a somewhat stronger way, as a little flow chart here, or a little program that would perform the calculations necessary to compute the transcript level of the endo-16 gene as a function of the different promoters. So that's clearly something both, let's say on the network level, we have many feedbacks and so on that generate what physicists would call nonlinearity. Also here we have a complexity that's both very difficult to disentangle experimentally, and then also difficult to treat in a mathematical computational framework. You can always simulate, you can run this, but does that give you an understanding of the phenomena? Does it tell you how sensitive certain parameters in the regulation of this gene are, compared to, on the behavior of the system, together with other parameters and so on? That's all getting very difficult. It's a lot easier in metabolic networks, but many questions that we're asking can just not be addressed exclusively at the level of metabolic networks, in which we can use linear algebra and so on, but they include these nonlinearities of this type. Second dimension, which I think is independent of the first one. So the network that I showed you, you can think of it as a gene regulatory network that happens on the low levels of the biological hierarchy of organization. Here I'm showing you all these different levels that include molecular levels, but also higher levels like pathways and modules at which we might be able to understand how an organism works, but also the cellular level and the higher levels, which clearly are impacted by the things that happen up here. And also, unlike engineering, it's the lower levels that are then shaped by evolution. Right. So all of these different levels are different levels at which you can look at your biological systems, but they're all connected in both ways, and it's not necessarily a simple relationship, as this example shows you. This is a regular fruit fly, lab animal for many geneticists. But a small mutation to this fruit fly will result in a fairly strong, I don't know how good this is to see, but this is a second thorax segment. And this second thorax segment is just a result of a tiny mutation in the genome of this fly, which means that, which is one example for a tiny change at one level of this hierarchy, which generates a fairly large change at another level. And there are many other changes, of course, that don't have these drastic consequences. It's just not a simple linear system that we would know the math of, that we know how to model a priori, maybe from other examples in physics, but it's more complicated than that. A third different aspect, I think that's again independent of the previous two ones. Everything in the previous two levels you could study in a single species, but as we know, many of us are involved in studies of different organisms, and they're important for several reasons. One is that they tell us about the dynamics of evolution, but also in many cases we're working with model systems that can tell us, for medical applications for instance, something about the biology of the human biology, but we can't do the experiment in humans, and so we do it in some other species. And then the question is, can we translate the results on this high level that we are usually aiming at? And then the evolution comes into play if we have several of these model systems that we can do experiments on. We need to then summarize the results, make inferences on human, or you can do the same thing with bacteria and so on. As we're getting more and more genome sequencing capacity, it's becoming more and more popular to look around what's going on. And then this tree pattern, this branching pattern is the scaffold on which we have to interpret a lot of data. And this is not something that we know as engineers how to do. That's not the classical structure that we would find in engineering. And again, I think it's an independent type of complexity that you have between different organisms, or even between different families. In genetics, you have complex relationship diagrams in a cohort that we have in Africa. We need to take these things into account, because otherwise we're making mistakes in the interpretation of genetic data and the associated phenotypes. And then I think there's a fourth, yet independent level at which we are faced with dynamics. And here's an example from the immune system, in which you have an immune cell here. Some don't ask me why, I used to know this. There's a little movie in this. Is this going? Yes. So this is a neutrophil surrounded by red blood cells. Yeah. And even if you know all the three dimensions that I showed you before, so evolution, the networks, the different levels, it would be difficult to understand that the dynamic behavior that these different levels and this different information is involved in leads to this phenotype, which is just that this neutrophil hunts the little Staph aureus bacterium that you see that's trying to escape. So while all the other things that I told you about they are complex by themselves and they give you some idea about a static image, I would argue that it's quite difficult to infer from these static ideas that this hunt happens in the bloodstream. And by the way, this is old technologies, a movie from the 50s. So in summary, I would submit, that's my postulate for today, that there are really these four different ways that are distinct and that are sort of, that that relate to different mathematical structures too, that together are present in many of the questions that we're asking using systems biology. Take your favorite problem that you're involved in. The problems that I've seen, often contain some of these levels, not all of them, but it's the combination of them that, in one way or another, we're often faced with. And it's the data that is generated in these 4 different dimensions. So each data point, each number, in my experience, can often be interpreted as something that you can describe using, if you want, coordinates. It's information about a particular gene at a particular level, or maybe a pathway. You have to think of gene here not as the absolute term, but as something that tells you which component of an organism. It could be on the higher levels a pathway or a cell which plays a role, or which cell type plays a role. Then we have the different species, or also different patients that may or may not be related. And we observe one given system that's configured like this across a particular time point. And there are, I think, many examples. I'm just listing a few. There's multifactorial diseases where the way that the different genes work together matters. There is the example that I mentioned where for clinical purposes, we take, instead of doing experiments in a human system, we take either an animal or we take a cellular system, and then we are faced with the challenge of translating the results. There's gene regulation, where we have different time points, for instance, in which we study the gene regulation. There's another example here where we use networks to see how these different genes then together influence each other. There's protein folding, which, for instance, that happens, on different time scales, and which represents one case in which the local behavior is difficult for us to extrapolate over a very long time. Like, the molecular moves happen in femtoseconds, and proteins fold on the order of seconds, and that poses a problem that you could call complexity. And I think that there's a corollary here to link this to visualization, which is that a visualization system that supports interaction with these 4 different dimensions in which we collect data, in which we try to come to global pictures and models and statistical analysis and so on, needs to support these 4 different dimensions in specific ways. And I think if that happened, I don't know of such a system, but I think if it happened, it would be a very powerful tool to address many of the questions that are being asked. That's not to say that all questions can be cast in this framework, but I think there are these 4, at least, principal dimensions in which a lot of the data is generated, a lot of the models are being built, and we have different visualization systems, including Bio.js, which support one or another of these aspects very nicely. Maybe there are different widgets also, or components of Bio.js that, and other systems that support one thing or the other thing. But here, really, even starting with the data lives in this higher dimensional space, and which are really independent dimensions. And I have not seen a system that lets us, browse and select and operate and model on these different subsets that you might want to look at there. So this was the sort of biology part, a suggestion for a visualization system. I don't know what these slides are doing here, but I wanted to talk about another problem, which is computational complexity. And I'm not going to talk about labels, but labels is one example very directly. In a visualization system, you're confronted with this problem that if you want to include the descriptions, textual descriptions of the nodes in your network, then you need to leave some space for that. And it's interesting that in most formulations of this problem on how you would like to draw this network, the resulting problem of leaving enough space for the labels and making things nice lead to NP-hard problems, which are problems that are not known how we address them in a scalable manner, so that graphs even of this size can be drawn efficiently, let alone interactively when you make changes on a screen quickly. So that's one problem directly in visualization. Another problem that I would like to spend a little bit more time on is this hotspot problem. I'm calling it like that because it's really a question that we get from biologists. Where is something going on in this network? And of course, there are these individual gene tests, like there are softwares like Limma and others that can test for differential expression. And there you can go gene by gene and say which one of these genes is differentially expressed, what are the p-values, for which of these genes do I have more evidence, less evidence. Then you can project that onto a network. But as this network paradigm that biology is organized in these networks is becoming more credible and more important, the question that many people are asking is not gene by gene, but sort of region-wise. Are there regions in which I can see more activity? Because these regions, even if we don't know now that they constitute a particular pathway, we can ask this question pathway by pathway. There's methods for that, but we would like to ask it in a network way. And you can use visualization for that. Like in this graph, I could use Bio.js or Cytoscape to put some colors on this graph and then just visually identify these regions. The problem with that is that it doesn't scale. Planar drawings are the ones that we want as nice visualization, which means that you avoid these intersections. But as soon as our network has any of these figures in it, any of the two, then you can no longer draw it in a nice way. But you end up with things like this, where it's a spaghetti hairball, and we need some help from computation to be able to identify these hotspots where there's something going on. Now, of course, if we say we want to identify a hotspot by a computer, we have to say what we mean by that. And one sort of almost ancient method that we developed a while ago is based on these values that are associated. So here I'm explaining a particular hotspot that could be a hotspot that would consist of 4 genes A, B, C, and D that interact. Think of it as transcriptomic data, with going up or down and so on, and associated p-values, which I'm expressing here as z-scores, which is an equivalent thing. Low significant p-value corresponds to high z-score here. So we're looking at the z-scores. The connection between z-scores is this, that a high z-score corresponds to a low p-value, which is the integral from the z-score to infinity under this normal distribution. But I'm not giving you a lecture about math today. Just let me summarize the method. The method works by summarizing the z-scores in a very, let's say, traditional way, by adding them up and then dividing by the square root of the number of things that we added. In the end, we want to compare hotspots that are small, consist only of a few proteins or genes. It doesn't actually matter where these numbers come from at this point, right? We're just considering this as a problem of interpreting these p-values or z-scores on a graph and trying to find regions in which we see a particular accumulation of significant ones, which in the z-scores again means high z-scores. So here's a classical way of summarizing them. It's actually, we chose the z-scores for this to be conveniently and quickly doable. Here we get a summary z-score for this one condition. The method that we formulated at the time works over several conditions, and basically here's an example. When you add all these numbers up and then divide by the square root of 4, you get this value. And so the method itself generalizes to several conditions, and then you sort, you bring the most significant summary statistic, which is here you have 2 times the 3, which is high. It adds up to 4 after dividing by the square root of the things that you add. and then we bring this in a sorted way, and then we have to correct for the fact that we're looking at 4 different conditions, because if I gave you many conditions at random, you would always find something that looks significant. So, but again, I'm not here to talk about the math of this. It's also not very complicated or ingenious. But what I want to say is that at the end, you will get a statistical measurement of how much this little potential pathway ABCD that's connected in the network could be a hotspot, yeah, from a statistical point of view. So there's a score here. And what I also wanted to say is that this work has been cited a lot, some 700, 800 times by now, and other people have derived methods from it. But we found out recently that there are 2 major issues with this. And they're kind of known, but we understand now better what is going on. So one issue is that you need, in order to find hotspots, you need to look at several of them. I cannot just a priori, I don't know that it's A, B, C, D, right? We have to look at all of them. So which means that we have to solve an optimization problem, which is, given all these values, and given that I have a way of computing a score for a given candidate, like A, B, C, D, can I find the best candidate in my network? And since this is, as we proved, there's also it's an NP-hard problem, we don't really have a good way to do this on these large graphs in a precise way. We had to have recourse to a general purpose heuristic, which basically does in a space of different solutions, where you modify one solution slightly to go to the other solution. You do kind of a walk, and many of you are familiar with this, and you try to reach the top. And of course, there's different issues with that. One issue is that you have, even if you get out of the city, you have these local maxima somewhere else where you can get stuck if you're just going up. And the other issue is that there are landscapes much worse than this, where there are many of them, so you're losing your time. And the thing is that we actually found that this is kind of the case. So in the Cytoscape plugin that exists for this, it's called J-Active Modules. Today, when you feed that with a network, we use the string database, a large network on the order of 500,000 edges. It actually takes a very long time. So this is 10 to the 6, 5 times 10 to the 6. And in terms of runtime on a standard workstation, it takes really, this is several days here. The default parameter, by the way, is still set at 2,500, which is roughly here. So if you run this 2,500 times, then the quality of the solution is this here. Whereas you would have to run it a lot longer, which is a problem by itself. So we are putting a warning on this plugin, which is very popular, that if you keep the default number of steps in which you search, you're not likely to find a really good solution, but basically a random solution, which is for us pretty bad, because we want to really not just publish nice papers, but also have an impact to biology and medicine and so on. So that's really a problem, and we don't know any other way to compute a good solution quickly right now. There's a second problem, which has to do with the fact that if we look at the different subnetworks that we visit, if we do this in a small simulated graph, we will come across many of these subnetworks. The scores that we will see are distributed like that, which is great. This is kind of how we built the statistic. I didn't tell you the whole story, but we made it so that when you do a long search, you're searching over a distribution that looks like this. But there is a second aspect to this, which is that if you look at small subnetworks, then you don't have so many of them. So the distributions the distributions over the ones that we're searching look similar, but in one case we're searching for the maximum of some thousands subnetworks for a small graph. In the other case, let's say if this is for 3 subnode networks, ABC, and this is for 5 node subnetworks in our simulation, the number of samples that we'll be looking at is a lot larger, and we want the maximum. So if you sample from a normal distribution and you sample much more often, you're bound to get a greater maximum the more often you sample. And this is something that had escaped us and had escaped many other people. It was kind of known that this plugin tends to get you large solutions, but we're not sure that people really understood the reason why that's the case. This is work of our graduate student, Irina. So which means that for larger, again, to summarize, larger subnetworks means you're taking many more samples, you're finding larger solutions, and if we do this many times, we see that the scores that we get from the higher networks in simulated data are higher scores, and it's only consistent with the way that this plugin works right now that you tend to get larger networks because it's the larger networks that have the higher scores, just because you can always find a larger one that has higher scores. This leads us now into, so there's two problems, computational complexity and the statistics that we need to fit. It's tricky to determine these distributions, at least empirically we cannot, because in order to do that we have to solve, each time we want to take a sample, we have to solve an NP-hard problem. So even if we did this only for one network, it's a bad thing, and we're looking into the theory of this, of extreme value distributions. It looks like this is the type of distributions that we have, but even to determine its parameters, it's a very hard problem. So at this point, we're stuck. We want to solve what we thought was a simple problem of highlighting in a complex graph hotspots for users, but it turns out to be a nasty problem statistically and computationally. So that inspired us to look at an alternative approach, which is at some level a little bit of a loss, because we restrict our solution space quite dramatically. We say, well, what happens if we don't allow arbitrary candidate networks, A, B, C, D, but what if we only allow stars like this, which consist of a central node in this network and then all the others. Still, visually, it would be impossible to draw large networks that you could do it by eye, but is there a computational way to do this? And we allow, you should know that not all of the neighbors here contribute a signal to this being a hotspot, right? Which would be maybe unrealistic, because now, if you look at the string database, for instance, you have so many interactions that come from so many different types that you would not expect a priori that in your pathway that is of interest in a particular analysis, all of the interactors would show some interesting feature like being differentially regulated, which is a low p-value that we then could pick up. So here in this sort of schematic example, we would just assume that a particular subset that we don't know a priori is affected and differentially regulated. So here's a quick example. Again, I'm not going through the math, but it's fairly straightforward. Here in this example, we would have 7 proteins, including the one in the middle, and 6 neighbors in the network. And we have different input scores for them. Think of them as p-values. And we sort, again, just akin to the statistics that I showed you earlier. We sort them and then determine the position in this list at which we can say that the first few of them together, when taken together, are less rare to occur in random data. That requires a little bit of statistic. It requires empirical distributions, but these empirical distributions are manageable because we don't need to search over a very large search space. In this case, we would then find of this statistic that evaluates how unlikely it is that in random data you would find the first few low p-values just by chance. We find then the minimum of that value, which here is 2 times 10 to the minus 9. But of course, we're doing this test many times for different subnetworks, and all of that is rolled into the empirical null distribution that we determine without larger problems on a normal computer in very little time. And then we tested this idea first on simulated data, where we planned some of these hotspots, just like we think of them, a region that has some some values that are lower, statistically low p-values. And there's a little algorithm that I'm not going to go into. We found that this works reasonably well, that we are able to detect better, even than other methods that you could tweak into this shape, for instance, GSEA, which some of you maybe know. You could tweak methods into that shape, but since we're doing the exact statistics, our ability to find these hotspots that we planted in simulated data is better, and certainly better than, in most cases, than just looking gene by gene, even if we introduce some noise and so on. But of course, more relevant, maybe, is that we also looked at gold standard data sets in which we know from biologists that there are known pathways that are induced by external stimuli to cells. Also, I'm not going to go into all the details, but the idea here was to compare the single gene approach to detecting hotspots or single genes that matter in these pathways that are stimulated versus the detection of these hotspots. And then what we did was we asked ourselves, well, if we have a set of experiments, in this our case 6, in which pairs of experiments are known to stimulate the same biological mechanisms from prior knowledge, can we reveal the similarity of these different stimulations by looking at what changes? We should be able, a method that is well able to pinpoint these hotspots should be able to show some overlap between, let's say, 2 experiments that stimulate the TNF pathway, or 2 experiments that stimulate the TGF-beta pathway, right? This is hard work to do. It's a lot of engineering and well, number crunching not, but it's a lot of piping together. We are happy with the answer that we get. We get a very clear improvement over detecting similarities in stimulations that are known to stimulate the same pathway that we're not getting from single gene analysis. Single gene analysis will point here and there, even if the 2 stimuli are known to be related, whereas we do get nice overlaps with this. And finally, also we're applying this to with our collaborators who helped us discuss and develop this idea to a genetic disease called cerebral cavernous malformations, which is a genetic disease. We know exactly what the genes are, but our collaborators would like to find out how does this play out in the transcriptome? How do the people in which these lesions happen in the brain and in the retina, how does it play out when the people have these genetic mutations that are exactly known, but how does it play out in development that these lesions form, and of course how could one counteract that? With LIMA we get for the 3 different genes, when you perturb them in mice, they have a very nice assay, very reproducible data. We're not getting any single overlap. You would hope that these 3 genes that by and large create the same disease, then also the way that they play out in the transcriptome there's some overlap, and then you can start looking there. There are some overlaps here, but nothing in the middle. With this approach to identify hotspots, we had one central gene that always showed up in all 3 analysis of these knockouts followed by transcriptomics. So in each case we had one hotspot that always come out. And then our collaborators believed enough in this hand waving that I've now done for you to actually test these things, and they tested in the mouse cerebrum. This is a human disease in the cerebrum. They found that this protein that I cannot, whose name I cannot tell you, that was unlikely for them, to their credit, they have tested it, forms very different structures in the cerebrum. It's kind of pointwise distributions in these veins. These are veins in the cerebrum, and they form long filaments. So to them it was totally unexpected. We don't know whether this has a causal impact on the disease, whether the lesions that you observe in people or in mice have anything to do with it, but it's a significant difference, and it's what we can expect in terms of finding a hotspot where do we see something significantly different. And they also did the test in the retina, and there we see the same thing. Here you see some points in green, which are the distribution of these proteins. And again, in the retina, they're forming these long filaments. This protein is known to form these filaments, but it's totally unexpected that it would change from a condition in which you have this gene active and you have it inactivated. So to summarize my talk, I tried to make links between visualization and complexity. In the first part, I've tried to suggest to you that maybe this complexity can be decomposed in different sources, networks, the hierarchy, the evolution between much of the data that we connect, and dynamics. They all, I think, have their particularities. They occur in a combined way in applications. We collect data in systems biology. A lot of the data can be placed somewhere in each one of these four contexts. And I think a visualization system would be nice, that would enable you to navigate in this space that is spanned by these four dimensions. In the second part, as you just seen, I talked about how we make compromises in visualization by finding hotspots that are maybe not our initial idea to have, any subgraph as a hotspot, but only these very specific star subgraphs. But maybe that's, good enough as a first cut. That's it. I need to just mention that the experimental work, the work on the stars in the end was done with our collaborators who work on this genetic disease. And there's people from the lab, ex-PhD student and another PhD student who was involved in that. And I look forward to hearing more about Bio.js and all the things around it, and welcome your questions now.
I'm really glad, Benno, that things are now beginning to work as expected. There's something I must say, because we had to kind of change the very last minute the keynote speaker who cancelled. So I had to ask last night, Benno, please, do you mind having your keynote early in the morning? Because, otherwise we're going to be a little bit screwed. So, yeah, it's we're hopefully back in track. Do we have any questions for Benno? Thomas.
So, actually, I really like that you dig into this active modules plugin, which I think it doesn't work optimally. So great that there's somebody going to make it work. I really appreciate the work. I was just wondering for this with this new star-like method, the downside you mentioned is that you fail to see the larger modules that may exist.
The one that are beyond star-like. So
right, beyond the first neighbors.
Any path of 3, let's say.
Right.
You cannot, any path larger than 3, you cannot necessarily capture.
Just an idea. Maybe you tried. Maybe it doesn't work. But would it make sense to still look 1 or 2 or 3 steps further in that star-like figure, but downway the further neighbors? So say they contribute less, and then maybe you will be able to kind of still have this compromise of still being able to see larger paths that are really strong and not.
That's a very good questions, and there are people who have tried things like that. There's 2 issues with that. Well, they're related. They're maybe the same thing. The thing is that these graphs, these networks, tend to be now so connected that there's a lot that you can do. Even if you go 2 steps, I didn't do that for a while, but even 2005 was the last time when I did that. I guess we went 3 steps, and then you get such a lot of possibilities that. I'm skeptical about the statistical aspect, which is that you're starting to pick up things that are not really connected. That's what I'm skeptical about, apart from the computational worries that I have, which it also means that you need to do a lot more work. And since we have no better solution to generating the statistical significance than to do permutation testing, it means that we do many, many times this, not only for the real data, but also for the permuted data. And but on the other hand, I've worked on problems before where I said, we can't do this, it's too complex computationally. And then 2 years later, or 3 years later, or somebody else said, you can do an algorithm like that. And so I think it's a suggestion that makes sense. Then the second methodological problem is to evaluate how well it works, because our world of pathways is still very small compared to what's out there. And I've shown you some attempts to validate this method, but the ones that are based on prior knowledge are somewhat satisfactory and they're good signals, though the experimental ones are very laborious. So I think this whole field of network biology, or extracting interesting networks, needs a lot more validation ideas on how we can validate findings.
We have one more question, I'm afraid.
So I'm not a biologist and I'm not a network scientist, so please excuse me if this question doesn't make any sense. But as I understand it, you're looking at these star topologies in order to simplify the underlying system to allow you to get to something that's algorithmically tractable. And then you pick this star, and statistically tractable. And so you pick this star system. Was there a motivation for that particular simplification? Was it arbitrary that you picked this star topology? Could you have gone for like triangles, or so like why was there some kind of like a priori reason why that's a good simplification, or was it just pragmatic?
Thanks. That's a good question. I haven't thought about this, but to me it seemed like a natural generalization of looking at a single protein. Look what's around it in a distance of one, while allowing to throw out some. I don't think it would work if you forced all the neighbors into this and said, I'm not believing that this is a good hotspot because many of the neighbors are not giving me a significant signal. But triangles are included, right? Because you have one node and it's two neighbors that could be picked up. On the other hand, that's not quite true, because if we look for many different topologies like triangles, but we're also looking for more, you pollute your statistics with other examples. So we include the triangles, but we include a lot of other things, and we pay a statistical price for it, which is detection power. So if all pathways were triangles, all the ones that would be interesting, then we would have that. But I don't honestly see, I could come up with immediate examples, even for a triangle. You could see that in protein complexes where you have three proteins that interact, but in these networks you have also gene regulatory interactions, and if you have a pathway A regulates B and B regulates C, then that's not necessarily a triangle, and we won't pick that up. But maybe we can come up with some ontology that would tell us what are the topologies that are interesting and so on. Still a little ways out, I think.
So we're gonna stop there. Thanks very much, Benno.